We're swimming in data these days, right? We have information coming at us from all sides. But here's a question. Does all that information actually lead to better, smarter decisions? Well, today, we're going to dig into that puzzle. We'll use the story of fighting a major crop disease to uncover a really powerful way of thinking, one that turns a bunch of separate signals into one clear, confident choice. And that really is the heart of the issue, isn't it? We've got more weather data, more tracking, more science than at any point in history. But just having all the pieces of a puzzle doesn't mean you automatically know how to put them together. The problem isn't that we don't have enough information, it's that we often lack a clear framework for how to actually use it. Okay, so to really get this, let's look at how things used to be done. For a long, long time, the fight against a disease called late blight, and this is a nasty one, it can wipe out entire fields, was guided by a pretty simple rule. That old rule was basically this: if the weather looked right for the disease to spread, you'd spray a fungicide. Makes total sense on the surface, right? It captures a really important part of the biology. But it's also kind of a blunt instrument. Favorable weather is just one clue. And if you act on that alone, you might end up spraying when you don't need to, or maybe even using the wrong tool for the job. And this is where things get really interesting. This is the big shift. Instead of just reacting to one single trigger, like the weather, we're moving to a much more structured logic, an integrated framework that combines multiple signals. This lets us build a way more accurate, way more actionable picture of what the real risk is. And this quote it absolutely nails it. The problem is not lack of data, it is lack of integration. The real challenge isn't just collecting more data points. It's about interpreting them in the right order, where each new piece of information actually changes the meaning of what came before it. So this integrated model organizes the decision around 4 key signals. The best way to think about it is like you're a detective building a case. Climate, that tells you if there's an opportunity for a crime. Inoculum, that tells you if the suspect is actually at the scene. The host, that's like checking the security system. And genotype, that's the detailed intel on the suspect's specific tactics. So let's break these 4 signals down. Let's decode these clues one by one and really see what they tell us. And more importantly, why the order you look at them in matters so much. First up, climate. This is our starting point. The right temperature, the right humidity, it all creates a window of opportunity for the disease. But, and this is so important, just because the window is open doesn't mean an intruder has actually come through it. Climate is a necessary condition, but it is not a confirmation of a threat. And get this is how powerful adding the next clue is. A 2025 study in Spain combined their weather data with actually monitoring the air for pathogen spores, that's the inoculum. And by adding just that one extra signal, their prediction accuracy for when the disease would actually show up jumped to over 90%. That's what integration gets you. And that brings us right to inoculum, our second signal. Honestly, this one is the real game changer. It tells you if the pathogen is actually there. It's the difference between knowing there's bad weather in the forecast and getting a notification that your specific flight has been canceled. Inoculum monitoring turns a vague, theoretical risk into an active, immediate threat. This idea is just so perfectly captured here. Inoculum distinguishes between climatic vulnerability and active pathogen exposure. In plain English, it stops you from taking action just because the weather's right when the actual threat isn't even in the area. Okay, on to our third signal, the host. You see, a plant isn't just resistant or not resistant in some kind of vacuum. Its defenses are, well, they're contextual. A plant might be super resistant to the pathogen strains in one area, but totally vulnerable to the ones just a few hundred miles away. And that leads us perfectly to our fourth and final signal, genotype. Think of this as getting specific intelligence on your adversary. Knowing the genetic makeup of the local pathogen tells you how it's going to behave. For example, a lineage in Europe called EU43 was found to be resistant to a major fungicide. If that's the dominant threat in your field, spraying that chemical would be a complete waste of time and money. So we have our 4 clues: climate, inoculum, host, and genotype. But the real breakthrough, the magic of this whole thing, isn't just having the clues. It's understanding how they connect, how they interact, and how they change each other's meaning when you look at them in the right order. So this is the crucial point. You don't just add these signals together and get a risk score. No, it's a logical flow. Each signal qualifies or refines the information you got from the one before it. It's a way of systematically cutting down on uncertainty by forcing all the variables into their proper biological relationship. So let's just walk through that sequence.
First, climate asks, is the window of opportunity even open? If the answer's no, you can stop right there. If it's yes, you move to inoculum. Is the threat actually at the door? If no, you just keep monitoring. If yes, you move to host. Okay, how strong are the locks on the door? And finally, genotype. Does this specific threat know how to pick these locks? See how each step gives you a much clearer picture?
And this table just shows it in action. If the weather's favorable but there's no inoculum detected, the smart move isn't to spray, it's to monitor more closely. If the weather is favorable and inoculum is present, but your crop is resistant, you might wait and observe. It's only when you have all three, favorable weather, a present pathogen, and a susceptible host, that a big intervention is clearly justified. It's so much more nuanced, right? So what all this really means is a huge shift for the folks making these decisions out in the field. It's a move away from just blindly following a rigid protocol and a move toward using informed biological judgment. This new logic just expands the entire toolkit. Your options are no longer just spray or don't spray. Now the right decision might be to wait and watch. Or it might be to spray right away but with a different chemical because of what you know about the genotype. Or maybe it's just to step back and reassess how vulnerable your crop truly is. And let's be really clear here, this is not about abandoning prevention or taking silly risks. Not at all. The goal here is to make every single intervention count. It's about improving the reason for acting, making sure that when you do something, it's for the right reason, at the right time, with exactly the right tool. This final quote just sums up the whole journey we've been on. It's a move away from fixed protocols and toward informed decisions. It's about being more adaptive, more precise, and in the end, just way more effective. You know, we spent this whole time talking about a plant disease, but this kind of logic applies almost anywhere. In a world that is just drowning in data, the real skill isn't finding more signals. It's learning how to read them together in the right sequence to build a coherent story. So I'll lead you with this. What complex problem in your world could you solve with a more integrated logic?